Welcome back to Jalen Bio. Today's module is on gas exchange. Diffusion and partial pressures of gases. Partial pressure represents a contribution of any gas in a mixture to the mixture's total pressure. Partial pressure is the pressure that's exerted by each gas within a mixture of gases and is measured in millimeters of mercury. It's written with a P followed with a symbol of the gas below it. Dalton's law states that the sum of all the partial pressure equals to the total atmospheric pressure. So in the first beaker, we have the oxygen pressure, 159 millimeters of mercury. The second beaker, we have nitrogen, 593 millimeters of mercury. And we combine them together, we have 752 millimeters of mercury. Gas diffusion between the air and the blood. Henry's law states that at a given temperature, the solubility of a gas in a liquid is dependent upon the partial pressure of the gas in the air and the solubility coefficient of the gas in the liquid. This value called the solubility coefficient is a volume of gas that dissolves in a specific volume of liquid at a given temperature and pressure. So CO2 has a greater solubility than oxygen, which is also then more soluble than nitrogen. Gases with a low solubility coefficient require a much more larger partial pressure gradient to push the gas into the liquid. So the amount of gas dissolved in a liquid is equal to the partial pressures times the solubility coefficient. This value will be given to you if there was any kind of calculations. The partial pressures of each gas inside a cell remains relatively constant except for during extreme strenuous activities. So the initial conditions are going to be right here, 100 mLs of plasma, 250 mLs of gas mixture with 50 mLs of oxygen, and 12.5 mLs of CO2. And then at equilibrium, you'll see that at 2000 mLs of gas mixture, you require 400 mLs of oxygen and 100 mLs of CO2 to maintain that balance. So some of the key points about diffusion and partial pressures are the equilibrium values for partial pressure are not the average of the initial partial pressures. The larger the partial pressure changes results from a greater percentage change of the gas volumes. And if a gas has a higher solubility coefficient, a greater volume will transfer in or out of the liquid. A higher solubility coefficient is associated with a higher rate of diffusion. This is very important to remember. And then the amount of gas in the liquid is most commonly expressed as equivalent volume that is transferred from a gas mixture and does not represent the actual additional volume added to the liquid. External respiration and internal respiration. External respiration occurs inside the lungs. Internal respiration occurs inside the tissue. In the lungs, oxygen moves from the air to the blood, whereas CO2 moves from the blood into the air. Whereas when it gets to the tissues, oxygen, O2, is going to move from the blood to the tissues, and CO2 moves in the opposite direction from the tissues to the blood. The oxygen shown in blue will diffuse towards the blood vessel and the blood cells, whereas the CO2 shown in red is going to exit out. And that is because you have 102 millimeters of mercury of oxygen on the alveoli, and inside the venous blood, it's going to have only 40 millimeters of mercury. So it goes from a high to low, 102 towards the 40. Whereas for CO2, you have 40 millimeters of mercury, and inside the venous blood, you have 45, so it goes the opposite direction. Then on the arterial side, you'll see it swaps over. The partial pressure of oxygen is 100 millimeters of mercury, whereas in the tissues, it's going to be 40, so it's going to want to go from the blood into the tissues, whereas CO2 is 40 millimeters of mercury versus 45, so CO2 is going to exit out. Just remember that the red arrows represent the diffusion of CO2, whereas the blue arrows represent diffusion of oxygen. Oxygen is CO2 transport in the blood. Hemoglobin transfers oxygen attached to an iron, CO2 can attach to the globin, and hydrogen ions can be also bound to the globin. So the red blood cells bind not only oxygen but also CO2 and hydrogen ions. The binding or release of oxygen, CO2, or hydrogen ions causes a conformational change in the hemoglobin that can temporarily alter the ability for hemoglobin to bind to any of the other substances. Hemoglobin can bind a maximum of 4 oxygen molecules. So the amount of oxygen bound to hemoglobin is expressed as a percentage of oxygen saturation of hemoglobin. As the partial pressure of oxygen increases, the hemoglobin saturation also increases, so more of it is available. The oxygen hemoglobin saturation curve is a graph that relates the partial pressure of oxygen and the percentage of oxygen saturation of hemoglobin. The oxygen hemoglobin saturation curve relates to the change in partial pressure of oxygen from about 20 millimeters of mercury to over 40 millimeters of mercury, which results in a large change in the hemoglobin saturation. While we progressively increase the amount of partial pressure of oxygen, there's going to be a less dramatic increase in saturation. So what you will notice is that hemoglobin tends to load oxygen as blood moves through the capillaries, moving about 98% saturation. Going to a higher altitude where the air is more thin and the partial pressure of oxygen decreases is going to be accompanied by a decrease in the partial pressure of alveolar oxygen, and therefore a decrease in the hemoglobin saturation. This is also why, in these situations, it could lead to altitude sickness, in which there's a decrease in alveolar partial pressure of oxygen that causes headache, nausea, difficulty sleeping, and even edema. So as oxygen binds to hemoglobin, it is easier for more oxygen to bind. As you can see over here, that if you go from increase of 20 to 40, the binding capacity saturation goes from 20 all the way to 80. However, as you get up to about 98, 99, in order to increase to the maximal 100, you would have to increase another 20 points of millimeters of mercury to increase 2 to 3%. As the oxygenated blood passes through our body during rest, only about 1/4 of all the oxygen is released from the hemoglobin and used by your systemic cells. There's also a concept called the oxygen reserve, which is the amount of oxygen that remains bound to hemoglobin even after passing through the systemic capillaries. Other variables that can influence hemoglobin's binding and release of oxygen include changes in temperature, changes in the pH, and as well as proton binding, and the presence of 2,3-BPG, also known as DPG, and CO2 binding to hemoglobin. Let's talk about some of these factors. The release of CO2 and protons from the globin causes a conformational change in the hemoglobin, which facilitates the bonding of oxygen to heme. The interaction between CO2 and protons is called the Bohr effect. An increase in body temperature will affect the hemoglobin, making it more flexible and making more oxygen able to be released at tissues. And this is referred to as the Haldane effect, when oxygen displaces CO2 from hemoglobin. And the last one is DPG. The chemical DPG, also known as 2,3-bisphosphoglycerate, is produced by red blood cells in an alternative glycolytic metabolic pathway and interferes with the binding of oxygen to hemoglobin. So factors that can decrease oxygen affinity for hemoglobin are going to be as follows: increasing temperature, increasing DPG, as well as decreasing pH. These will cause the oxygen curve to shift right. Anything that increases pH, decreases 2,3-DPG, and decreases temperature will cause it to shift left. Factors that shift it to the left means that it increases oxygen affinity for hemoglobin, whereas if it shifts to the right, it decreases oxygen affinity for hemoglobin. CO2 transport. Carbon dioxide is transported in the blood from the systemic cells to the alveoli through dissolving in the plasma, about 7%. Attached to the globin portion is about 23% in the hemoglobin, and bicarbonate is also dissolved in 70%. So majority is going to be in the bicarbonate form, also known as HCO3-. The Haldane effect states that CO2 binds a lot more easily whenever the hemoglobin is unoxygenated. Gas exchange between blood and tissues. At systemic cells, carbon dioxide diffuses into the blood due to lower partial pressure of CO2 in the blood, and diffuses into the erythrocytes or red blood cells to form bicarbonate, which keeps the pCO2 very low. The bicarbonate ion will then diffuse out of the erythrocyte back into the blood plasma with the accompanying movement of the chloride ion. This helps keep the electrical neutrality in this process, referred to as a chloride shift. So CO2 diffuses from the tissues in towards the blood capillaries. As CO2 comes in, CO2 is going to be bound to the red blood cell, interacts with water, and becomes carbonic acid. It will then quickly dissociate into protons and bicarbonate. The protons will bind onto the red blood cells and the hemoglobin, and then the bicarbonate gets expelled out to the blood plasma when the chloride ions go in. So it maintains negative charge on both sides. When the blood reaches the pulmonary capillaries, the bicarbonate will then move back into the erythrocyte, and the chloride will begin to move out, reacting with the protons to reform carbonic acid with the help of an enzyme called carbonic anhydrase, which will reconvert CO2 and water and diffuse into the blood and then to the alveoli because of the partial pressure CO2 gradient. So right here you can see the bicarbonate is going to go back in to the red blood cell and chloride comes out. The bicarbonate is going to reform with the protons, creating carbonic acid, and really quickly with the help of carbonic anhydrase enzyme, create CO2 and water. CO2 and water will then be diffused across the alveoli, and now the CO2 gets to go out because of the gradient. Control of breathing. The skeletal muscles of the breathing are innervated by the somatic motor neurons, and they're rhythmically coordinated by the nuclei in the brain stem. The rate and depth of breathing are altered through reflexes that are consciously controlled at the cerebrum and the brain stem. Different kinds of receptors can pick up changes. Chemoreceptors can monitor the fluctuations of concentrations of hydrogen ions and the concentration of respiratory gases inside the cerebrospinal fluid. We also have the central chemoreceptors that are located inside the medulla oblongata, and they can pick up changes in the protons, CO2 will diffuse quickly from the blood into CSF under the carbonic anhydrase and can catalyze to CO2 in water and can be reformed into protons in bicarbonate. There's also peripheral chemoreceptors inside the carotid arteries and also the aortic arch that can also monitor both protons and partial pressure of CO2. So we have the conscious control, emotional control, the stretching of the lungs, and these guys can all affect the brain stem, and the medulla and the pons. So the glossopharyngeal nerve and the vagus nerve can send signals towards the brain to change and alter the way we breathe, based on sensors inside the brain stem, carotid bodies, and aortic arch. The phrenic nerves and the intercostal nerves will then send signals back out to the muscles of the diaphragm and intercostals and external costals to change the way you breathe. So some of the key variables are the amount of partial pressure of oxygen, partial pressure of CO2, and the pH inside the CSF. It should be noted that there are many different parts of the brain, including the hypothalamus, that can increase the breathing rate when the body is warmer and decrease it whenever the body is colder. The emotional states are controlled by our limbic system that can trigger memory and changes to your breathing. And of course, the frontal lobe and the cerebral cortex can also change the patterns of breathing when you're talking, singing, or holding your breath. The respiratory center contains patterns of generating neurons with intrinsic depolarizing capacity. Let's go over some of these key centers inside the brain stem. APC stands for the apopneutic center. PTC stands for pneumotaxic center. The APC provides stimulatory input, and then the pneumotaxic provides inhibitory input. So you have a yes signal, and then you also have a no signal. Inside the pons and medulla, we also have the DRG, the dorsal respiratory group, that responds to the blood CO2 and oxygen. And these guys can affect the ability of DRG and change the way they breathe. New research has shown that there is also a retrotrapezoid nucleus, which receives information from the chemoreceptors inside the brainstem and lungs that can affect the VRG. Some other areas that should be noted include the caudal VRG, pre-Botzinger area, and the Botzinger, which can then be also used to maintain rhythms in breathing.